Have you ever just stopped and wondered how a single cell in your body knows exactly which page to read from your massive genetic library? I mean, it's this incredibly precise molecular dance, all about switching genes on and off at just the right time. So today, let's dive into the absolutely elegant world of this cellular machinery.
I mean, really think about that for a second. Your DNA has billions of letters of code, right? But there's no index, no table of contents. So how in the world does a cell find that one specific sentence, a single gene it needs to read to make a particular protein? It's a navigational challenge that's just mind-boggling.
All right, to get to the bottom of this, we need to meet the hero of our story, an enzyme called RNA Polymerase. But you know, every hero has a weakness, and this one has a huge blind spot that really sets up the whole problem of reading our genes.
And here's where things get really interesting. See, in simpler organisms like bacteria, their polymerase is a real jack-of-all-trades. It finds the gene, it copies it, it does it all by itself. But in our cells, our polymerase is more like a Formula One race car. It's unbelievably powerful and fast, but it needs a whole pit crew just to get it to the starting line. Without that help, it's totally lost.
So, to make this whole thing easier to picture, let's use an analogy. Let's pretend we're shooting a huge movie. Our RNA polymerase, that's our super expensive, high-tech camera. And the DNA is this massive, sprawling landscape we have to film in. Now, you can't just plop that camera down anywhere and start rolling. You need a crew to find the perfect spot and set up the shot.
So the big question is, how does the crew find out where to set up? Well, it all starts with them looking for a very specific landmark on that huge DNA landscape. They're looking for the X on the genetic map that basically screams, "Start here!"
And that landmark is this short, very specific sequence of DNA letters called the Tata box. And yeah, it's pretty much what it sounds like, a string of T's and A's. This little sequence is the first signpost that says, hey, a gene starts just a little ways down the road.
Okay, so the very first member of our film crew to spot this signpost is our location scout. It's a protein with the perfect name, the Tata binding protein, or just TBP for short. Its one and only job is to find that Tata box and grab on tight.
And the way it does this is just so cool. When scientists first figured out its structure, they saw that TBP is literally shaped like a saddle. It just sits right down on top of the DNA molecule, gripping it like a rider on a horse.
But here is where it gets really clever. TBP doesn't just sit there. The act of binding forces the DNA to bend, and I mean really bend. It creates this sharp kink that acts like a massive physical beacon, basically like raising a giant flag for the rest of the crew to see.
And we're not talking about some gentle little curve here. This is a sharp 80-degree bend. Think about that. This major distortion of the DNA's perfect double helix is the critical, hey, over here, signal that gets the whole assembly line going. Okay, so the flag is up. Now what? Well, once that 80-degree bend is in place, the rest of the film crew, a whole group of proteins we call general transcription factors, starts showing up to build a launch platform for our camera. And this whole thing happens in a very precise order, kind of like building IKEA furniture. First, the main anchor, a big complex called TFID, lands on that bent DNA. Then, another protein, TFIB, comes in and acts as a bridge, making the perfect docking spot. After that, another factor arrives, basically as a special escort, bringing our polymerase camera right to the location. And finally, the last few pieces snap into place, and boom, you've got a complete launch platform. Now, let's zoom in on that bridge protein, TFIB, for a second. It's not just a simple connector. You should think of it more like a high-precision clamp on a camera tripod. Its job is to measure the distance from that Tata box just right so that it positions the polymerase perfectly. It makes sure its lens, its active site, is pointed at the exact first letter of the gene that needs to be copied.
So the whole platform is built. The camera is in position, it's aimed correctly, but nothing's happening. The scene is set, but we need a director to finally yell, action.
And that director role is played by the very last protein to join the party, TFIIH. It has two absolutely critical jobs to do to finally get this camera rolling and start transcription. Job number one, open up the script. The DNA is a double helix, right? Two strands twisted together. Well, TFIH has this built-in function that acts like a molecular zipper. It uses a bit of chemical energy to pry open those two DNA strands right at the start of the gene, creating a little bubble and exposing the letters that need to be read. Job number two, give the polymerase the push it needs to actually start moving. It does this by adding some phosphate groups onto a long, flexible tail that hangs off the polymerase. This chemical modification acts like flipping a switch, breaking the polymerase's connection to the launch pad and literally kicking it into gear.
And with that, we have liftoff. The DNA script is open, the polymerase camera is free and moving, and it starts cruising down the DNA strand, reading the code and building a matching RNA molecule. The movie is finally rolling.
But you know, what if the polymerase messes up a line? Just like in filmmaking, you need some quality control to make sure the final take is perfect. And of course, the cell has a clever plan for that too.
Now, the polymerase is incredibly accurate, but it's not perfect. Every once in a while, it'll add the wrong RNA letter to the growing chain. When that happens, it can get jammed. It basically stalls out on the DNA track. This is something called transcription arrest.
This is where our script supervisor steps onto the set. It's another protein, this one called TFIIS. Its whole job is to constantly scan for these stalled-out polymerases, and when it finds one, it rushes in to the rescue.
And the way TFIS fixes the problem is it's beautiful. It doesn't fix the mistake itself. Instead, it activates a hidden ability that's been dormant inside the polymerase all along, a kind of self-correction mode. TFIS essentially taps the polymerase on the shoulder and says, hey, back up, snip out that last bit with the error, and give it another shot. It's an incredibly elegant proofreading system. So, let's just quickly recap the whole thing. Starting transcription isn't a solo act. It's a true team effort. The entire process is this tightly choreographed sequence. You find the spot, you assemble the platform, you launch the camera, and you even proofread the take. And it all relies on these amazing physical signals, like that incredible 80-degree bend in the DNA. It's this whole complex system that makes sure your genetic blueprint is read with amazing accuracy, every single time.
And just take a moment to let this sink in. This intricate, multi-step dance we just walked through, it's happening millions of times every single second, inside trillions of your cells right now, just to keep you going. It really makes you wonder, doesn't it? What other amazing cellular machines are humming away inside of us, performing their own silent, essential miracles?